We are continuing in chapter 8, and we talked in general about the species concept and the idea that the biological species concept is not very useful for most people, because even if you are looking at, say, a fish or an invertebrate, particularly an invertebrate, that does have sexual reproduction, you don't know what it's reproducing with sexually, and most people aren't going to go out and do the experiment and put those together. So a more functional definition of species is whatever the taxonomist defined as a species at that particular time, and then you base it off of the keys that you would use for invertebrates or fishes or algae. And off of molecular methods, which are becoming more and more common. And one of the most common molecular methods is using ribosomal RNA for taxonomic identification of organisms. Because this has become such an important method for many species of organisms, and particularly for microbes where they don't have sexual reproduction and their morphology is relatively invariant. I mean, there's just a lot of sort of, maybe a few different types of morphologies, particularly in the bacteria and archaea. They use ribosomal RNA molecules for identification of organisms. This is an image of a ribosomal RNA molecule. And ribosomal RNA forms part of the core of the ribosomes and is present in all organisms, except for if you include viruses as organisms and they don't have them. It has sort of two general functions, and one is parts of it are catalytic. That is, they're actually a reactive, it's a reactive molecule, and other parts are structural in the ribosome. Can anybody remind me what the function of ribosomes is in the cell?
They transcribe proteins.
Exactly. They take messenger RNA and turn that signal and that code into a protein. Good. All cells have to do that, right? So it's absolutely essential that it works. For that reason, there are parts of the molecule that are very conserved. And in this particular schematic of a ribosomal RNA, you can see these red areas that tend to be fairly conserved. The reason for that is that if there are mutations in those areas, the molecule becomes inoperational, and it's not able to either have the correct structure or the correct enzymatic activity. In which case, the organism that has that mutation dies and cannot reproduce, right? So there's a very hard selection for not changing the base pairs in those particular parts of the molecule. There's other parts of the molecule that are not so important. So one of the things we see these hairpins, right? We see going around and coming back, right? So that makes a structure here. And it's possible that these bits out here are really not very important, that all they are there for is just as spacers to get it back around and create that stem of where it comes back and base pairs to itself. In that case, there are mutations in these particular base pairs, and I'm not sure if this is a highly variable portion of the molecule or not, but there are areas where there's just not much function to those particular base pairs. You can get fairly rapid evolution and change in the sequence of that particular section of the molecule. So this leads us to having a molecule that has enough information in it to quite a few base pairs you can see here. So you can have enough information where mutations can give you a big differentiation between species. And that is, all of the organisms. You can look at the deepest differences, and you could like at domains of organisms, or you can look at fairly shallow differences like genera or species, maybe even. In the extreme case, you can see that there's the bacterium that is responsible for Swiss cheese. You can actually, if you get one small mutation, it doesn't make propionic acid, which gives the Swiss cheese its flavor. You can also find that in the ribosomal RNA. There's one base pair difference, and so they can do a rapid test to figure out if the bacteria they're using to create that cheese allows for the appropriate flavor to develop. What this says is that there's a variable clock speed in ribosomal RNA. So if you are going to look for differences, say at the kingdom level or phylum level or something like that, would you look in the highly variable or the highly invariable regions of the molecule? Does anybody have an idea? Invariable, good. I'll give that one to Abigail and take it away from Cassie. Right, there has to be some variation, right? And so you definitely need to see some variation. And the areas that there's no variation are areas what we call useful for universal primers. Because when we use the molecular methodology to basically expand the number of molecules, we need some way to sequence it and some way to amplify the number of copies. And to do that, you need to be able to have a series of bases that you know are there, and then you can use those to start synthesizing and creating multiple copies of the molecule, which then you can sequence. There are certain sections that all organisms have the same base pairs. And this then allows you to create a universal primer that will work on a bacteria, on an archaea, on eukarya. But there are other parts. There are other parts of the molecules that are somewhat variable, slightly variable, right? And so those are not going to change very much over evolutionary time. That most archaea will have one sequence and most bacteria will have another sequence, for example. So at a very deep phylogenetic level, you can find those differences. In contrast, if you have an area that's changing very rapidly, that's a fast clock area. And that is one that you could use to see the differences between genera, say, or maybe even species, right? Because there's not hard selection to stop mutation from occurring there. And so just over random changes in time, there's going to be drift in the sequences, and they're not going to be the same, even in organisms that are relatively closely related to each other. This makes it an incredibly useful molecule because you can zoom in on the sequence in some parts and use it for big differences. And you can zoom in on the sequence in other parts and use it for very fine taxonomic differences. For some reason, students have a lot of trouble understanding this concept. And every time I ask a question on an exam related to the fast clock and slow clock portions and how they're useful in taxonomy and species identification and understanding phylogeny, students tend to get it wrong. Some get it, but others don't. I think part of that is because the typical student that's in here has not done a lot of molecular taxonomy. But even when I teach microbiology and have students that have a heavy molecular background, they've not thought about these issues of evolution and needing to conserve certain molecules and others and variable clock speeds and evolution in molecular taxonomy. Some species are under a lot stricter selection than others as well. So this next molecule is Thermus aquaticus, and this is a obligate thermophilic organism. And thermophilic organisms tend to have less variability in the ribosomal RNA than less thermophilic. Let me see something real quick. Okay. The reason for this is related to stability, right? So if we have a organism that's living in a very hot environment like Thermus aquaticus, almost boiling water, what's going to happen to the structure of the proteins and the ribosomes and such? they'll unfold or break apart, great. They'll tend to denature, right? So if they denature and they do unfold and break apart, then the organism's not going to be able to survive. So it has to have a way to get past that. That's why when you throw us in boiling water, we're toast, right? But these organisms can live in almost boiling water. And we'll talk about this in extreme environments. We'll go back to this. In this particular case, this molecule, though, you're going to have these areas where it comes back in base pairs with itself, right? So here, purple, not variable. Here, it's base pairing with itself. Here, most of these purple areas are in areas where they're coming back in base pairing with themselves. These tend to be much less variable in this particular group of organisms than in ones that are found in bacteria that are found at more normal temperatures experienced in aquatic environments. So this is under very, very heavy selective pressure to have very stable and unchanging ribosomal proteins. So the clock speed can vary both with the type of molecule, and it can also vary with the amount of with the importance of the location of the molecule and then the selective pressure that the organisms are under that have that molecule. You can imagine those that are in really cold environments might be able to have fairly loose ones because they don't have to work as hard to maintain their structure, the psychrophilic organisms. Okay, so we've got two questions here. You can answer either one of these questions if you want to, but this is one of the questions of the day. And I should mention that if I ask several questions of the day and somebody is gone, then I tend to not pay attention to the fact that they answered one. I try to make sure they answered all the questions of the day that I have. Okay, we've got some good answers here. How do variable rates of change in the RNA molecule relate to areas crucial to the function of the molecule? The more crucial it is to the function of the molecule, the slower the rate of change is, right? Because evolution, well, there'll be hard selection, and evolution will not allow for changes in those because the organism basically can't make it more. How do areas with variable rates of change in the rRNA molecule make it useful for taxonomic comparisons across broad taxonomic lines of more divergent organisms? So the slow clock areas, the parts that don't change very much, can be used to make broad taxonomic characterizations. The fast clock areas, the ones with high variability that are not essential to the function of the molecule, those can be used to make comparisons of recently diverged organisms. Then the other part of the question that I added on to this is how can you calibrate the clock, right? And so we do know that how long ago groups of organisms diverged. How do we know that? What evidence we use to figure out how long ago groups of organisms diverged? Say birds from dinosaurs or birds from lizards.
Do we just carbon date their remains?
Yes, we can do that for relatively recently diverged organisms because carbon dating is about 14,000, 20,000 years or something like that. But there's some of them are millions of years ago. DNA doesn't exactly work for that. How do we know what the history of evolution is for the most part of organisms, and when they became distinct from each other?
Maybe transition species like that plant you mentioned the other day, Cara.
Right. So what you can do is you can go back in what record and figure out when the land plants showed up. What's the name of that record that we used to figure that out? Fossil record. Good, Aaron. Got it. Okay, yeah, fossils. We can use fossils. We can go back and we can use not necessarily carbon for very recent things, but other isotopes can be used to date those fossils, and then the different layers over geological time and then their order, then we know when a new species shows up in the fossil record and the first progenitor of a species, when the first dog shows up or something like that. Then we can say, okay, we've got these two fishes that are different from each other, and the first time in the fossil record that shows them varying is 1 million years ago. And then you can look at the amount of change in the ribosomal RNA and say, this is how we can calibrate the clock, because we knew there were 10 changes over 10 million years or something like that. So we can calibrate that clock and get an idea of how variable the DNA, the ribosomal RNA is. You could do that with other molecules as well. So that's how we use that. Expect a question on this, and if you've got any questions on it, you need to ask me about it, because it's sort of a foundational approach to taxonomy that people are taking now. Here is a ribosomal RNA tree of the eukaryotes. And as I mentioned, they are the high-level organization of how we see the differences between organisms has changed drastically. So for example, the colored flagellates were considered protists and lumped with all this stuff down here. Yet we are very closely related to the choanoflagellates. And we're, as I mentioned before, we're also quite closely related to the fungi. The plants, everything down here of these branches, you can't really, they're no more or less, it's not well resolved down here, but they're the least related groups to each other. And then we'll see some of the organisms we're going to talk about, the amoeba, the dinoflagellates, and so these are some of the algae. These are some of the other algae that we have. Giardia is way off of here. So we're gonna use this. We can use this to take it a step further, the molecular stuff to take it a step further than morphology, and really tease out what the evolution is of these organisms and phylogeny of these organisms is. And here's another version of that, a relatively recent, as of a couple of years ago, version of that type of tree with more detail. And we can see that some of the algae show up here, but some of them show up down here. So the protists have been really separated out into a lot of groups, and there's a lot of diversity out there that we kind of lump because we think that different people look very different from each other, even though we're the same species. Yet our tendency is to not notice the difference between different microbes, just because of one, because they have less differentiation, and two, we're not keyed into doing that. We can also classify organisms by functional significance. So they play roles in the environment, and those are not necessarily lined up to taxonomy. So you can think of organisms being autotrophic or heterotrophic, self-feeding or other feeding. Autotrophs use carbon dioxide to build organic carbon, and they need some external source of energy to do so. But there's algae that can do that. There are plants that can do that. And we'll also discuss there are bacteria, chemoautotrophic bacteria, so they're not very related at all, yet they're all bringing carbon into food webs. Heterotrophs range from simple bacteria to us, right? But we play similar roles. We can play similar roles in that we are ingesting organic carbon and breaking it down. So the next point was the chemoautotrophs versus the photoautotrophs. We can talk about, I just mentioned that, where they get their energy to fix carbon. Detritivity versus predation. Are you eating live things or are you eating dead things? Another area we can classify organisms as. When we get into streams, we have functional feeding groups of invertebrates. The shredders shred leaves, grazers eat algae or plants, collectors collect particles, carnivores eat other animals. These are not taxonomically based, but from an ecological point of view, they could be useful. You could have for example, one primary producer replace another one and not have a huge difference in the food web, even if the primary producers are not very closely related to each other. We can also classify organisms how they interact with each other, and these are the types of direct interactions that organisms can have. And we'll be talking about this throughout the rest of the class, and you need to know this terminology so you're not lost. And some of it's very straightforward. Competition is two negative interactions. Mutualism is a reciprocal positive interaction between two organisms. Some people call this symbiosis. I think that is an incorrect use of terminology, because symbio means organisms living together or close to each other. And there are many organisms that live close to each other that do not have positive interactions on each other. For example, a parasite, right, has a negative impact on its host, yet they live very close together. So they are symbiotic, they're just not mutualistic. What some people call predation, I tend to call exploitation, and that's because there are other interactions where positive and negative interactions occur. So for an example of, predation is fairly straightforward. Something eats a plant, it hurts the plant, and it helps the animal that eats the plant. But we just talked about a parasite, right, that lives inside. Maybe that hurts its host. Maybe there's an epiphyte that lives on a macrophyte. So the algae outcompetes it for light, but the plant gives it someplace to live. And so the macrophyte has a positive effect on the algae that's growing on the surface of it, the epiphyte, but there's a negative effect on the plant. Many interactions probably have no effect. That there's commensalism is a +0. One organism benefits, the other one, it doesn't have much effect on it. So you might think of something like, I don't know, a dung beetle or something like that, where the animal that releases a dung, it's gone. That doesn't matter to it, but it has a big effect on the dung beetle. Amensalism, some organisms have no effect on another, but the other organism has a very negative effect on it. Probably the most common interaction is neutralism, that most species do not directly interact with each other. If you have an invertebrate in the stream, it may not interact at all with the microbe that's breaking down the leaf, or it might, but many species do not interact with each other directly. Indirectly, everyone's linked together, and we'll talk about that as well. Another question for you: what is the ultimate amensalous species on Earth that negatively affects more species than any others, yet most species do not have any effect one way or the other on it? Yeah, everybody's getting it now. Humans. So that was lazy, Cassie. You just use the up arrow to show the answer above. Good one. Okay, yeah, we're having a negative effect on a lot of species. We do have some positive effect on some species, rats, cockroaches, right? And they may not have a huge negative effect on us. We have our mutualisms, right? What are the big mutualisms we have, humans? Organisms that would not make it if were it not for us. Yeah, livestock, right? The cattle can maybe not be so adapted to living. For example, here, bison can make it through the winter, cattle can't without being fed. Cropland, pandas have become that way. Yeah, so there's quite a few of those. We can also classify by habitat, and I think you need to look at table 8.3 and spend some time making sure you know those words, because I will throw out words like epiphyte, right? Lives on plants, right? That's one that maybe some of you don't remember, but I'll talk about organisms that are benthic, that are found in the bottom of streams or rivers. So there's terms in there that you will need in the future, and I'm going to throw them out there, and you can stop me and ask me to define them, but the more times you look at them, the better off you'll be.